Monoclonal antibodies. Monoclonal antibodies are protein molecules made in the laboratory from hybridoma cells. These are the terms related to monoclonal antibodies. Mono means one and clonal means clone antibodies. Antibodies is a protein produced by the body's immune system when it attacks harmful substances, called antigen is a substance that when introduced into the body stimulates the production of an antibody. Epitope is the part of the antigen that the antibody binds. B cell is a type of white blood cell and specifically, a type of lymphocyte. Myeloma cell is a type of cancer that develops from cells in the bone marrow called plasma cells. Hybridoma is a hybrid cell made in the laboratory by using abnormal cell with a cancer cell, usually a myeloma or lymphoma. Introduction to monoclonal antibodies.
An antigen, such as a protein, has several different sites that can react with different antibodies. Monoclonal antibodies are antibodies that react with only one particular site on an antigen, called an epitope. Consequently, they are highly specific and have applications in diagnostic microbiology and cancer therapy. The first step is to inoculate an animal with the antigen carrying the specific epitope. B cells are then isolated from the spleen and then mixed with a type of cancer cell called a myeloma cell that grows continuously. The two types of cells fuse together to form cells called hybridomas. The mixture of cells is cultured under conditions which permit growth of the hybridoma cells, but not the B cells or the myeloma cells. The hybridoma cells are then separated into individual chambers and tested for ability to produce the desired antibody. The hybridoma cells producing the desired monoclonal antibody are then cultured, and monoclonal antibodies are isolated and purified.
Monoclonal antibodies to treat acute lymphocytic leukemia. Antibodies are proteins made by the body's immune system to help fight infections. Man-made versions, called monoclonal antibodies, can be designed to attack a specific target, such as a substance on the surface of lymphocytes. Blinatumomab is a special kind of monoclonal antibody because it can attach to two different proteins at the same time. One part of blinatumomab attaches to the CD19 protein, which is found on B cells, including some leukemia and lymphoma cells. Another part attaches to CD3, a protein found on immune cells called T cells. By binding to both of these proteins, this drug brings the cancer cells and immune cells together, which is thought to cause the immune system to attack the cancer cells. Monoclonal antibodies have been applied clinically to the diagnosis and therapy of an array of human disorders, including cancer and infectious diseases, and have been used for the modulation